So my name is Mushri, and I'm here to present to you a short update on our pilot project that the Desmoid Tumor Foundation funded and is also co-led by Candace in the audience here, and as well as Joelle, Strayella, Dave Sherman, and Aaron Thorner. And I have no conflicts to tell you about. So our focus is on the spontaneous regression of desmoid tumors, and particularly the super general research questions that are motivating us is whether there are extrinsic or intrinsic signals that are controlling this behavior that are responsible for spontaneous regression. And if we can understand them, then could we perhaps use that to control an aggressive desmoid tumor to induce a more aggressive state, or at least understand whether a patient would require a more aggressive disease management or not. Now, in studying desmoid tumors, I started studying them since I was a graduate student in Dr. Benjamin Allman's lab in Toronto. And one of the most consistent things that I've always been challenged with is that desmoid tumors exist as a co-mixture of both the actual desmoid tumor cells as well as fibroblastic cells that do not carry the beta-catenin or the APC mutations. And so it makes studying them in vitro super challenging, as well as any other type of bulk readout is also very challenging to look at. This was also the same challenge when I was moved to the Broad Institute under Jesse Bowen's supervision as postdoc to generate new desmoid tumor cell lines. Again, the biggest problem we run into is that fibroblasts take over. The desmoid tumor cells aren't really growing. And as you heard, these cultures that are pure desmoid tumor cells are very slow and very tough to work with in vitro. But we do know that in the majority of patients, they do carry these point mutations, and as we alluded to, there are very specific mutations in desmoid tumor patients, namely in the beta-catenin, the T41A and S45F are by far the most predominant residues that you'll see mutants. So it's very important in a lot of the studies that we do in vitro and also in situ is to always capture the mutation. Otherwise, we'll probably be, following perhaps, false paths because we're not really understanding whether the signal we're finding is coming from desmoid tumor cells or stromal cells. So whether in culture, in trying to track the impact of media conditions or drug conditions on desmoid tumor cultures, we invented ways to capture and quantify the beta-catenin allelic fractions, as well as in single-cell RNA sequencing. We had to basically adapt methodologies that allow us to not only generate the single-cell profile, but also combine it with the genotype of single cells so that we can understand better what the real signals are. So with those sort of brief backgrounds in looking into this planning project about spontaneous regression, we approach it with the hypothesis that we can detect differences transcriptionally between regressing and non-regressing desmoid tumors. But in order for us to test this hypothesis, it was critical that we combine it with our ability to concurrently genotype the cells to understand better what the signals are. So very early pilot in the very beginning with just 3 tissues from the biobank at Dana-Farber. One desmoid tumor patient sample with T41A mutation, one with an S45F, and then the adjacent normal from that latter sample. And as expected, the single cell, or in this case, single nuclei data you generate, you get this beautiful UMAP. You see some cells clustering in different clusters. Some of them come from different patients. Some of them are patient-specific or even sample-specific. But we don't really know what these cells are. We can use cell type annotations. It tells you that there are macrophages, skeletal muscle cells, endothelial cells, et cetera. And then everything else is considered a stromal cell because we lack the reference to tell us what the desmoid cells are. So everything shows up as a fibroblast or a stromal cell. So we looked into the raw sequencing data that is generated by this methodology, namely the 10x single nuclei RNA-seq. And we looked at the raw sequences around the beta-catenin hotspot region for these very, very early samples. And we saw what we expected to see, that in the desmoid tumor tissues, we see the mutations for beta-catenin, specifically the T41A that we expected for sample 1, the S45F for sample 2, and then in the adjacent normal, we see it as rather blank wild-type beta-catenin readout. So this suggests that there is specificity in this approach that we're taking, albeit it's only a small minority of cells that we are able to annotate in this manner, because you're typically biased towards the end of the transcript when you're doing single-cell RNA-seq, and this wasn't, long-read sequencing methodology, but we can still capture some information about beta-catenin, and we see that these cells that we can annotate with beta-catenin status occupy different clusters in the transcriptional space. So from there, we expanded the cohort to the planning project cohort. So we expanded it to 15 samples from 12 patients, some of them from spontaneously regressing cases, some of them from progressing cases, and others are considered stable. And we collected the majority of tumor samples, but also some adjacent muscle samples as sort of a reference control. And in total, we collected about over 180,000 single nuclei that passed our QC. They occupy a number of different transcriptional clusters. So despite what we typically talk about in terms of the uniformity of the genetics of beta-catenin, they occupy a lot of heterogeneity, both between patients and then within specific tumors. And then there's also a lot of clusters that are shared among patients. Usually this would indicate that these are normal or stromal populations. So our focus is on, for this specific talk, is in how can we capture differences between regressing and progressing tumors. The first question that we looked at is whether there are differences in cellular composition. And we see that at least there are trends suggesting that the regressing tumors tend to have more endothelial cell populations, although we cannot really rule out the fact that regressing tumors might be smaller or maybe more extracellular matrix than cellular samples, and therefore we're looking at the effect of regression rather than a cause of it. But at least the early data suggests that perhaps some cellular compositional differences exist between progressing and regressing cases. But more importantly, we wanted to look at the beta-catenin status of these single nuclei. Again, we performed this transcriptional methodology of looking at the raw sequencing, finding the beta-catenin hotspot region, and assigning a genotype status to every single nuclei. Again, a very small minority of the nuclei could be annotated in this fashion, only less than 4%. But we got around 1,000, a little bit under 1,000 muted nuclei that we can at least begin to understand whether there are any transcriptional differences. How do we increase this? Well, one way would be the direct amplification of the beta-catenin, in addition to the amplification of the unique cell barcode, so we can attach the identity of the single nuclei or the single cell to the beta-catenin genotype. This is what we've been working on in my lab. And we've tested this with cell lines so that we can remove the confounders of mixtures of cells existing in a sample, and also because the desmoid tumor samples are so precious that we want to optimize the protocol in samples where you have abundance of RNA. So we tested this in 2 cell lines, the A549 that is wild type for beta-catenin and LS180 that has the S45F mutation in beta-catenin. And doing a direct amplification is possible and does increase our capacity to annotate more cells. And this is something that we would hope to continue working on with the cohort of desmoid tumor samples that we already have. Another way of doing this is through inference. So we collected around 800, 900 mutant cells with a beta-catenin genotype in them. And we also collected around 2,000 cells that we can confidently call wild type, because beta-catenin is a heterozygous mutation, and so just finding any wild type read is not a sure way of saying that that cell is a wild type for beta-catenin. So from this small set of single nuclei, we can generate a gene expression signature that tells us something about the mutation status of beta-catenin, and we then can apply it to what we call the ambiguous or no information single nuclei. And from this, we can at least see hints that we can generate a gene expression signature, and we can apply the signature on unknown cells so that we can score them for their beta-catenin mutational status, and then assign them a class, whether it's a mutant cell or a wild-type cell. And what we can appreciate from this, looking at the UMAP with the red and green clusters on the far right, is that the mutant cells occupy distinct transcriptional clusters. And therefore, despite having perhaps the same mutation on the genetic level, the transcriptional states are different, and this is after batch correction as well. So we wanted to go back to the question of spontaneous regression. Can we take only the mutant cells, or at least what we infer to be a mutant, beta-catenin mutant cell, and compare them in terms of their clinical behavior or the behavior that the tumor they were derived from? And the answer is we can find some differences, some gene expression differences between mutant cells that come from regressing tumors and mutant cells that come from progressing tumors. It's still very early data. It's only very few cells that we're talking about. However, at least there is, our data suggests that it's possible to do this, and it gives us a more zoomed in look at not just the composition on the cellular level, but the same cells that carry the same mutations, how their transcriptional states are different. And from this sort of exercise, we found at least one hit, collagen 25A1, that seems to be correlative with progression status or spontaneous regressing. Again, this is specifically in the cells that we are labeling as beta-catenin mutant. And as Ben is not here, but he would always tell me after a result like this, well, is this true, and unrelated? And the answer is we don't know yet. This is still super early to know whether this is just a correlation or whether there is some mechanistic effect. But what we do know, and I apologize if the picture looks a little bit blurry, but we do know that at least it's not one patient driving this relationship, that at least we see about 3 samples that are progressing and about 3 samples that are regressing. The COL25A1 differences appear to be consistent between the two, suggestive that perhaps, in summary, that the concurrent genotyping of beta-catenin and also in the future APC in single cell and single nuclei atlases of desmoid tumors is essential, that we can perhaps identify cellular compositional differences between regressing and progressing tumors. And specifically within the mutant population, perhaps that there are transcriptional differences between progressing and spontaneously regressing tumors, that perhaps we can look at these differences and identify biomarkers to predict tumor behavior, and biomarkers or mechanistic hypotheses that we can then go to the lab, modulate or work in vivo, like Chris will talk about in a sec, modulate in vivo to see whether or not they have an impact on the tumor behavior. So with that, we want to make all of our data possible, explorable by the community. So we're building a web portal to make all of the data easier to share. We're still super early here, but my hope, and this is the reality with working on a rare disease like desmoid tumors, is that we need to be very inventive in the way that we study the disease and inventive in the way that we share data. Because as you see from all these talks, applying the same tools that already existed for more common diseases don't always work for desmoids. And so if there's anything that taught me, if I learned anything from researching this disease, is that you're constantly thinking about inventing a new tool to help the study. And so we want to build a portal to allow for folks, to go onto your phone and search for things about desmoid tumors all in one place. And with that, I want to thank everyone. I wish to thank Candace, Joelle, and Dave for their leadership on this project. Also, our funding sources in the Desmoid Tumor Foundation in particular have been funding our work since I was a graduate student, and now I'm happy to say I have my own group in MIT. We're the high throughput sciences facility, but we also do desmoid tumor research as well. And without the Desmoid Tumor Foundation, I don't think I would have been able to get to where I am today. So I'm very thankful for everybody here, and thank you to the patients for donating their tissues. Thank you. Any questions?
I have a quick question. In your 15 samples, I know it was only 2, but it looked like there were 2 pediatric samples.
Yep.
So you probably can't comment too much, but I was obviously curious if there's any difference in signature between the 2. There's some of us who do this that think that it may be a more aggressive disease in children compared to adults, and we don't have any biological data to kind of support that other than just our clinical observations. But obviously, enriching the population for younger patients would be a wonderful way to be able to look and compare so that we can make decisions, potentially differently based on the age of a patient.
I 100% agree. I haven't looked at pediatric and how the regression versus progression, or even just in general, transcriptional signatures between pediatric and adult desmoids might look like. I'm happy to dig deeper into that, and we can talk later, but absolutely. I think by increasing the cohort and having more examples of that, we ensure that what we're looking at is not being driven by that 1 pediatric or 2 pediatric cases. Yes.
First, great data. That's extraordinary. Thank you. I think it shows the power of technology and things we can learn from really amazing pathology with dedicated single-cell studies. On your study, a lot of data, so I had trouble keep all the. Did you see a difference in the TME between the regressors or the progressors? I mean, just in terms of the different populations of the cells on the single-cell level, I couldn't tell.
Yeah, good question. I haven't looked beyond the sort of, at a very general level, the cellular composition. I haven't really looked deeper into whether there's specific tumor microenvironment populations that are very specifically different. The endothelial population sort of pops out right away, but it's really hard to say with only the 15 patients. As much as 180,000 cells sounds like a lot, it's not really much when we're looking at, after teasing out all of the what's mutant and what's not, it becomes a little bit of a problem.
So often with single cell, you can get pathway mapping. Have you looked at that at all in this data set? So are there pathways that are activated you wouldn't have anticipated that it could be beta-catenin or not?
Yeah, I looked specifically. So when you're limited by how many cells, so like around 800 cells or so, you're limited by the statistical power for something to pop up as different. Right. So instead of doing an unbiased way and say, let's look at all the hallmarks or all of the gene signatures, the only approach really that you'd be statistically powered for is to say, I have a hypothesis that is beta-catenin, and then you go in and look at it. So I have that hypothesis, and it's not that. So it would be more of a candidate gene or candidate pathway approach that we should go about doing this.
And there was one last question. There was one slide you showed the regressors. There was like a big green box on that one. There's one slide. Is that the COL25A?
This one?
Go back. Go back. Go back. Go back. Go back. Go back. Almost there. This one here. So, yes. So I'm red-green colorblind, so I apologize for red-green colors. But
no problem.
In the regret, what is that?
This?
Yeah.
These are the endothelial.
Those the endothelial cells. Okay. Yeah. So just as a cell type. That's all. It's endothelial.
Correct. And it's not even, a granular look at the cell type. This is a very sort of shallow way of dividing the cells into different categories. I'd imagine, something a little bit more granular gives us more exciting hypotheses, but at the same time, you could quickly lose your statistical power to say anything about it.
Okay, thank you very much. Great talk.
Yes.
Yeah, fantastic talk, and congrats with your route into independence. So that's really great to see.
That's awesome.
But yeah, So just wondering, like the classical Wnt targets, like Exin-2, they don't allow you to discriminate the tumor cells from the stromal cells?
Yeah, so I don't find, so if you ask anybody, you know, what are the classic targets of beta-catenin, everybody will give you Exin-2, and then it becomes sort of very different from everybody's perspective. I do not know exactly the technical or mechanistic reason for why Exin-2 is not a good gene to look at in single cell data, but it just is. It's a very sparse sort of readout for Exin-2. So that leaves things like DKK2 can show up, DKK1 can show up, and I believe it did show up in one of the volcano plots that we looked at. There's a DKK2 on the left side. But yeah, like in general, looking at the classic beta-catenin targets doesn't really tell us a whole lot. I don't know if it's because we're specifically looking at mutant cells, so they're all having beta-catenin classic targets up anyway. My personal inclination is more towards the question of why specific residues in desmoid tumors are found and not others, that there's the canonical classic way of thinking about Wnt, and then there's a desmoid-specific slice, and I think we still haven't mapped that out yet.
Now that you found Col25A1 is up, what do you plan to do with that? What's your next step to evaluate the association between that and desmoids?
So as a basic biologist, the first thing I want to do is modulate it and see what happens in culture. As a sanity check, we want to stain for it in IHC in tissues and see how that, maybe perhaps there is a spatial pattern that could tell us something about the expression, but absolutely, a functional study is important. I envision a lot of collaborations with folks that are studying it in vivo would be very important because, as you noted, these cells are so hard to study in vitro as well.
Got a question to the patients with, or the tissues from regressing patients.
Yes.
Was there really biopsy during spontaneous regression, or did you have the biopsy beforehand and the tumor regressed later?
Yeah, that's a great question for Candace, actually.
Because who would biopsy a regressing tumor?
They were actually all surgical resection specimens. So these were from...
So you resect regressing desmoids?
Our biobank is started in 2005. Actually, a lot of the samples were collected in the era of doing surgery more frequently. But yeah, so they're good chunks of tissue, and they're from tumors that were regressing, and we really meticulously, obsessively went into the charts of each patient to know what the behavior of the tumor was around the time of surgery.
I have a second question on the so-called wild type. What's the driver mutation of a wild type?
Yeah.
I think I saw it on one of the slides, or?
Yeah, I don't think, so at least going back to my experience in culture is that these are stromal fibroblasts like any other solid tissue. The only difference is that in, let's say, colorectal cancer, breast cancer, lung cancer, it's easy to distinguish them phenotypically. In desmoid tumors, it's super hard. Now, the issue with desmoid tumors, by just flagging something as wild type, is that it's a beta-catenin heterozygous mutation. In single cell data, it's super sparse. So you may get like 2 or 3 reads of beta-catenin, and just by luck alone, all 3 would be wild type. You can't really conclude that that cell is wild type. So therefore, we look at, it's a bit of statistics, but you're basically looking at the 95th percentile of, how many reads can you really see in a cell? And if you still see it as wild type, then you're going to have to draw a line and say, okay, this cell is wild type. I don't think that this cell, we don't know, but I don't think that this particular cell would be the tumor causing cell. It's strictly a stromal cell that perhaps is communicating with a tumor, helping modulate the behavior of a tumor, but not necessarily, the initiator of the tumor per se. At least that's my thinking.
You don't think they harbor an APC mutation?
So, yeah, so aside from those 2 patients with the known APC mutation, the others where you see a beta-catenin somatic mutation in them, it's highly unlikely that you have a mixture of APC cells as well as beta.
That's true. There's either the one or the other.
Correct.
Yeah. Thank you.
Thank you. Yes.
In terms of, so we have sort of a binary, progressing or regressing, but was there any work done to assess that in more quantitative terms? Kinetics?
Yeah, in terms of the annotations I had, or at least for the small cohort, we wanted to do, and it's definitely a very challenging thing to say, can we be more quantitative about that? Again, one of the biggest issues is that maybe 15 sounds like a large number. It's a tiny, tiny number when you're trying to do statistics with it, and then it becomes, a very big problem. I suspect with a bigger cohort that we can have better power to say, maybe there is, different types of regressions or different velocities or accelerations of a tumor, and then use that. But I don't think we've done that per se for this cohort yet. But it's a great question, and I'm excited to, get even more and more granular with this type of study. All righty, thank you.